Synthetic biology is a new field bringing together engineers, physicists, molecular biology, and chemists with the goal of using engineering principles to put together biomolecular components, genes, proteins, other bits of DNA, into novel biological circuits and pathways, and to use these networks and pathways to reprogram organisms, endowing them with novel functions to address a range of applications that include health, energy, food, and the environment. The field really grew out of interest in the human genome effort. In about the mid-90s, as the Human Genome Project was beginning to present large parts lists for different organisms, the leaders of that field recognized that really one of the next challenges was going to be to figure out how these parts operate in networks and pathways in these organisms, given the recognition that it's really these networks and pathways that underlie interesting biology and disease. The challenge with doing that was that we really didn't have the data and the technology in order to so-called reverse engineer or infer the structure of natural networks. Many engineers, including myself and folks in my lab, were excited nonetheless to get involved with genomics, involved in molecular biology. And so instead of thinking about reverse engineering or inferring natural networks, we thought about what engineers typically do, which is could we tinker? Could we forward engineer and put together networks that in this case were wet and not dry in the sense of electrical engineer? And with Tim Gardner, one of my students at the time, he and I helped to launch the field with this approach, wherein we sat back and said, could we now come up with a circuit schematic just as an electrical engineer would, model it mathematically to get a sense of how it might function, and then go find the parts that would be the biological equivalent of electronic components that could make up that circuit, and then go in using molecular biology techniques to piece together those parts into plasmids or DNA constructs, get it into a cell and see if it functions as such. Tim and I cycle through over about 9 months many different circuits to consider, and what we decided to focus on to start was a genetic toggle switch. This was motivated by electrical engineering, wherein there are toggle switches or flip-flops or RS latches. A toggle switch in electrical engineering is a simple form of memory. It's a very simple circuit that can be flipped between 0, 1 or on-off state by the transient delivery of a pulse. Say electrical pulse or might be light. Our machines that we carry around, iPhones, iPads, PCs, are made up of millions, if not billions, of such toggle switches. Tim and I sat back and said, okay, how would we do this in the case inside a cell, inside a bacterium? And the schematic we came up with was quite simple. We basically had 2 interacting genes that were set up so that they each wanted to be on. So they were driven by so-called constitutive promoters. A promoter is an on switch for a gene. It's a strip of DNA. But we set it up so it's a circuit so that the protein produced for gene A wants to bind to the on switch for gene B, shutting it off. And the protein produced by gene B wants to bind to the on switch for gene A, shutting it off. So each wants to be on, but each is trying to shut each other off. It's a mutually inhibitory network. Well, in principle, you can set up this circuit so that it wants to exist in one of 2 stable states. Either state A, gene A is on, gene B is off, or state B, gene B is on, gene A is off. And in principle, you can actually flip it between these states by transiently delivering a chemical or environmental stimulus that will shut off the currently active gene. So let's say, for example, you're in state A. If you can deliver a chemical that will temporarily inactivate gene A or inactivate its protein, well, provided that chemical is there for a suitable amount of time, now gene B that had wanted to be on but was kept off by gene A can now come on, produce its protein, and once it reaches a high enough concentration, it can shut off gene A, and you can remove the chemical that was keeping gene A now off from the system, and you flip the system from state A, gene A is on, gene B is off, to state B, gene B is on, gene A is off. And this was the general concept. And with Tim and myself, we initially started as many in those days, so this is around 1999, with mathematical modeling. And so we were able to mathematical model that process and convince ourselves it could work. And then with Charles Cantor, who was a bioengineering colleague of ours here at Boston University, he led us into his lab. Tim learned enough molecular biology and genetic engineering techniques to actually create this in E. coli. And he created one that could respond to 2 different chemicals and one that could respond to chemical and heat shock. And in a matter of 9 months, Tim was talented enough as a bioengineer to actually get functioning toggle switches behaving as bistable fashion inside E. coli. And this, along with, interestingly, a very related effort done by Michael Ilowitz and Stan Liebler, where they designed a repressilating oscillating circuit that now had 3 genes. Gene A would try to shut off gene B, which would try to shut off gene C, which would try to shut off gene A. And in principle, that's a ring oscillator, wherein you should have a blinking circuit, for example. They constructed that in E. coli. And these papers were published back to back in January 2000 in Nature, and really helped to serve to catalyze the field of synthetic biology. Well, you can imagine now that you've got a circuit that can endow a cell with memory. And this actually helped inspire a number of people on the biocomputing side, where they realized, well, now you could maybe program a cell just as you could, albeit in a simpler fashion, a circuit. And whilst there was a lot of interest in biocomputing, to think of it as replacing our electronic circuits inside our PCs is probably not the right way to think about it. But thinking about programming these cells to create, for example, now programmable cells to do a range of functions is, in fact, the right way to think about it, and a dominant theme in synthetic biology. So, for example, we've used toggle switches to create whole cell biosensors that enable us now to program bugs that could sense, for example, heavy metals like lead, or nasty chemicals like a DNA damaging chemical, or a pathogen. In this case, now you could put them in the environment, or you could put them inside somebody's bodies, or have them at checkpoints for imports to see, does your imported toy have lead paint? Do you have an anthrax outbreak outside of a government building? And the beauty of the toggle switch is that it now can restore memory, stores it in memory so that you can see, yes, I had an event. We similarly now have used related switches that are based on RNA that enable us now to turn multiple genes on and off dynamically inside a cell as a means to redirect the metabolic flux inside that cell. And we're now working with a number of biotech companies to see how you could use that to increase the efficiency of engineered organisms that are being used, for example, to convert biomass into different energy sources, such as fuel, which would include diesel, ethanol, butanol. We are also very, very excited about how you can use synthetic biology to engineer organisms to address a range of therapeutic applications. So, for example, we've engineered bacteriophage to go after bacterial biofilms. Biofilms are communities of bacteria that are attached to a surface. This includes plaque on your teeth, this includes gunk in your sink, this includes stuff on the side of a ship. Why a bioengineer like myself is interested in biofilms is that bugs inside biofilms are 1,000 times more resistant to antibiotics than bugs that are free swimming. Further is that anytime you put something inside somebody's body, like an artificial knee or an artificial hip or a cardiac pacemaker or a defibrillator or a brain stimulator, the number one risk is now not the general surgery involved in the operation, it's that the person's going to get a biofilm infection. We took on this challenge and said, how could we do this? So we looked at engineering bacteriophage. Bacteriophage are viruses that only go after bacteria. And we engineered them so that when they went and entered bacteria in biofilms, they would go through a lytic phase, wherein they're going to make multiple copies of themselves, harnessing the machinery of that cell, and then burst open the cell, and now you have hundreds, millions of progeny that can go after additional bugs. The challenge there is that you can't get below that initial layer of the biofilm. And so we further engineered these bacteriophage so that they can actually degrade the biofilm and expose more and more bugs. And what we were able to show was that with these engineered phage, we could get 99.99%, greater than that, clearance of the biofilm, whether on a medical implant or in an industrial setting. And my student Tim Liu, who was the leader of that, along with another student, Mike Curris, looked to actually move this into the commercial space. And initially focused on the therapeutics. They were encouraged then to look at industrial applications. It turns out any kind of machine that is exposed to water or living organisms can often get bedeviled by biofilms. So air conditioning systems have problems with biofilms. Air gas, oil gas pipelines, paper mills. And they initially were focusing on using engineered phage to eliminate those biofilms. But that turned out to be a very high regulatory challenge. And so they then shifted focus to now using engineered phage to detect pathogens in the food industry and in hospitals. And their goal, which I think they're just about ready to hit, is to detect as few as 10 bacterial cells in under an hour for under $10. we're excited about moving further is toward additional infectious disease applications. And now with support from the Gates Foundation, we're engineering probiotics to sense and respond to different infections. So for example, in our case, we're engineering lactobacilli to respond to cholera infections. So here we've engineered them so that they would respond to 2 different signals from cholera. And in that response, they would produce an antimicrobial peptide specific to cholera. And the beauty of this is cholera drugs are very expensive and can be highly toxic. Now, in principle, and we're just starting work in vitro on this, you could spike this into somebody's yogurt in the face of a cholera outbreak, for example, in Haiti, just after the earthquake, or put it in a pill. And now this is so much cheaper than developing the drug itself and much less toxic. The only individuals who will see the drug will be those that are actually exposed to cholera. And so I think we'll see in the coming decade or 2 major advances in synthetic biology that are going to impact our lives, whether it's on energy, food, through protein nutraceuticals, health along the lines I said, or even environment, where you could see these coming in to help clear up different problems in different sites. I think some of the most intriguing scientific questions are really trying to understand how the natural networks are wired up and how they function. And why I bring that up is I think we have much to learn from these organisms that have evolved over millions, in some cases billions of years, to create these circuits and networks that do pretty complicated functions, in some cases under very hostile environments. And so I think synthetic biology, while I focus primarily on the applications, holds a lot of promise on the basic science side of enabling us to use these as tools to gain insight into how the bugs actually are using these networks to do interesting and more complicated functions.